Review units 3.1 and 3.2 about DNA, the structure of DNA, and how it replicates. First off, we're going to talk about the people. First person was Rosalind Franklin, and she was able to take an X-ray photo of DNA, which proved it was two strands in a helix shape, opposite direction to each of the strands. So they were running anti-parallel to one another. And she also proved that the sugar and phosphate backbone was to the outside and that bases were to the inside. So she was able to make those conclusions. Unfortunately, she did not publish those things. Maurice Wilkins did. He was also a British scientist. He published those results and won a Nobel Prize for it. Watson and Crick took that information and built the first model of DNA. And in doing so, published that and were also awarded a Nobel Prize. Now, Chargaff was an important scientist also that helped them understand that the number of adenines and thymines were equal to the amount to each other, and the amount of cytosines and guanines were always equal to each other in living things. So this led them to understand about base pairing rules. So we know that guanine is always matched up with cytosine, and that adenine is always matched up with thymine. So then we get into some more of the structural components of DNA. First off, DNA is a deoxyribonucleic acid, and that means it's made up of deoxyribose sugar, a phosphate group, and a base. These three pieces combined are called a nucleotide. When you get multiple nucleotides connected to one another, it creates a nucleic acid. So we see these pieces fitting together like puzzle pieces. Again, we have base pairs where guanine is matched up with cytosine, thymine with adenine, cytosine with guanine, and the pattern continues in an anti-parallel fashion, meaning one with sugar first and one with the phosphate first. So when we see this structure, this is our classic double helix strand that we would see made up of individual nucleotides that are bonded to one another. So once we have the structure of DNA, we're going to move over to this strand, we can look at how it's replicated. And this was something that Watson and Crick, when they made their model, were very concerned about. And they were concerned about this fact because we knew that when cells were going to divide, we needed to have a copy of the DNA for every cell. It has all the instructions on how the cell is going to function, work, what it's supposed to do. So we need to make sure that every cell has those instructions. So in order to make a copy or go through DNA replication, there are 3 steps to this that we discussed in class. The first step is that an enzyme called DNA helicase will come in and break the hydrogen bonds that hold the bases together. So the bases start to pull apart as helicase works itself up the strand. So helicase comes in and breaks the 2 strands apart. Now, as helicase does this, a second enzyme shows up on the scene, and that is DNA polymerase. DNA polymerase has a couple jobs. The first job that it's going to do is make sure that base pairs match up properly. So it's going to make sure that across from a guanine, we get a cytosine. And on the other half of the strand, over here, we're going to have a guanine matching up with the cytosine. The polymerase continues to work on that strand. Following the helicase. So as the helicase makes its way up the strand, breaking it, the polymerase follows behind. It's making sure that guanines and cytosines match up, cytosine with guanine. Now we have an adenine, so it will make sure a thymine is there. If we have a thymine, it'll make sure an adenine matches up there. So this process continues up the strand of DNA. And as we continue across from the cytosine, we get a guanine. Across from this guanine, we get a cytosine, the polymerase making sure that is happening all along the way. The polymerase can also proofread and go back. So if for some reason one base decides to jump ahead and the wrong one gets matched up, the polymerase will be able to come through and double check and make sure that that is corrected so that there are no errors to that DNA strand. So the polymerase continues to work up the strands. Until we have created two completely new strands. Now these two strands are identical to each other, but they are also identical to the original strand that we had here on the table. Now the cell can go through mitosis and divide in half, with half going to the one cell and half going to the other. This works because we used the original strand on both sides, and we just picked complementary bases that went with them. We made sure we always matched up our base pairs. Hope you have found this helpful, and you can do all of the recovery pieces and parts so that you can take your retest.